So my involvement in the development of gene therapy actually preceded that important moment at the turn of the century. It actually was in the mid-'80s when I began my research in developing the concept of treating patients with genetic diseases using gene therapy. The key point then was the notion of using a virus as a way to deliver genes to cells of patients that have rare genetic diseases. This then progressed pretty rapidly, actually, to around in early 1990 era, where the first human trials of gene therapy occurred. Again, we were also part of that in the development of potential treatments for cystic fibrosis. Actually, at the turn of the century, the field stalled a bit because there were some adverse events, and the technology that we had developed was not adequate to deliver the gene safely into the right cells. So that first 10 years of the century was a period where there was a lot of science, but not a lot of clinical translation. That changed right around the 2010, and since then, dramatic examples of gene therapy helping patients. Children born without sight can now see. Patients that can clot with hemophilia can now participate in their normal lives. And probably one of the most miraculous examples was the use of a gene therapy vector that came out of our lab to treat patients with an inherited form of ALS called spinal muscular atrophy. These children should otherwise die within the first 18 months of age, and now they're living, talking, and walking. Approaching gene therapy now was a lot easier than when I first began to think about it. First of all, we know what all of our genes are, and because of sophisticated techniques of sequencing, we could identify genetic defects in any one of us by simply sequencing our own genome. So we really know the genetic basis for virtually any disease. The question is, how do you leverage that towards treatment? And I think the biggest challenge now and when we began is delivery. That is, delivering the gene safely and efficiently to the right cells. We've made tremendous progress, in particular, a family of viruses that we discovered at the turn of the century called adeno-associated viruses, or AAV, that were much better than anything we had before and have served as the foundation of the current field in delivering genes into the eye for blindness, liver, muscle, heart, and the brain. But we have a long ways to go to be able to do that with a kind of efficiency that would be necessary to maximize the impact of gene therapy broadly across many of the 7,000 rare diseases. We are really in a remarkable era. I began my training as a physician, seeing patients with rare genetic diseases. And it struck me at the time that all we could do was to diagnose the diseases that are often disabling or lethal and advise the families to go and to take their loved one home and to enjoy whatever time that they have left with them. That was our specialty in medicine. My dream had been that at some point we could develop treatment so that when those young kids are diagnosed, that they have a future, not just to go home and to die of their disease. We are at the beginning of this revolution. There are approved gene therapy treatments that are curing diseases, but it's only the beginning. It is a new day. Unfortunately, though, that there are many diseases that are still untreatable, and our hope is we can leverage the experiences that we've generated to date across many, many other diseases. It is a race against time, and time is probably the most precious commodity for many of these families and their loved ones. But progress is being made at breathtaking speed. Developing gene therapies for rare diseases is challenging scientifically, but as we've been talking, is now feasible, and we see a path forward. Maybe the biggest challenge ultimately will be is whether these approaches are commercially viable. In other words, is it possible that we can encourage the biopharmaceutical industry to embrace the development of these products and to develop them, manufacture the products, and distribute them? The challenge is that they're cures. We call it one and done, one-time injection, and potentially eliminate the disease, which creates pricing problems because most drugs one has to take for your lifetime, and with a chronic genetic disease, that would mean many, many different administrations of the treatment. The other problem is many of these diseases are very rare. So the combination of cures, single treatments with very rare diseases makes it difficult for biopharmaceutical companies to actually justify the investment of these treatments. And in the few examples when they have, they charge very, very high costs for the treatments. So high that it really prohibits access globally, and in particular in Latin America. This is a challenge, but we're convinced it's going to change. We are exploring other models other than standard commercial models that would allow the development of these treatments and access. And our belief is that any patient, independent of how rare their disease is or how much money they have, should have access to these treatments. I look forward to another 10 years of research in gene therapy, in which we maintain a pretty active involvement, pretty excited about the direction we're going and where we're headed. Clearly, there will be a number of other cures or treatments of these rare diseases that will leverage the experiences that we've had to date. And so over 10 years, there will be many, many more diseases, many, many more families, many more individuals living with these diseases that will benefit from them using the technology that we have now. And I'm quite gratified with that and hope to see that as a physician. But as a scientist, I'm even more excited about the opportunities to make gene therapy more successful and safer by improving delivery, improving safety, and deploying some of these more innovative cutting-edge technologies to modifying our genes, such as something you may have heard of, genome editing or CRISPR, in which we may be able to actually modify our genome to correct a gene defect that would be forever part of that patient.